looks like we're starting to get things filled up, so we'll kick things off. Thanks so much for joining us tonight. My name is Glenn Marangelo, and I am the Development Director and one of the founding board members for the Missoula Butterfly House and Insectarium. And if you haven't noticed yet, we're trying something a little different this evening. Boy, for years people have been saying, hey, can you video the lectures so we can watch them at some time. And we've had a little bit going on over the last couple of years, so this was something that was a little bit low on the priority list. But tonight's a little different. So thanks to our friends at MCAT, tonight's lecture is being recorded by the Missoula Community Access Television as part of a media assistance grant donated to the Missoula Butterfly House and Insectarium by MCAT. So thank you so much. And over the years, we have had so many amazing lectures, and looking down the road, our hope is to be able to start recording in one fashion or another most of our lectures, to have them available as a resource on our website. Beverages not included with those. But so be patient, we'll see where this ends up going, but we hope to continue this moving forward. So now that the new year is here, the school field trip season is getting into full swing, and just about every day that we're open, we're hosting at least one, if not two, field trips for schools all across western Montana. And since my office is kind of right across from the classroom, and I'm typically chained at my desk doing some kind of fundraising task, I usually hear when the field trips are wrapping up. And I hear all the kids in unison say, thank you, which made me think of tonight. Because tonight, as with every night that we've held this lecture over the last 10 years, DraftWorks has generously donated their delicious beer, and now their beer and the hard seltzer, for Bugs Brews. So in so many ways, they have been incredible partners over the years, not just their beverage donations, but their enthusiasm and their support for our buggy mission. And I don't know if anybody from DraftWorks is here tonight. There was a couple people that were potentially on their way. But just like all of those really well-mannered children at the end of a field trip, I'd like you to all join me in saying thank you to DraftWorks. All right, that was awesome. All right, and for those of you that may not know the tradition, I usually do a bug joke or two as part of our introduction. So the first one, can a cricket jump higher than a building?
No, buildings can't jump.
That wasn't somebody I told a joke to earlier. Oh. The answer was no, buildings can't jump. All right. No, I'm pretty sure you have not heard this one. This one may test your vocabulary at the same time. So people who cannot distinguish between etymology and entomology bug me in ways that I cannot put into words. And just as an aside, I tested the joke out on Jen, our executive director, where she's probably still talking to people, and she said on a resume for one of our positions, someone said, I really love etymology. It's like, okay, that one goes to the side. Okay. Before we dive into things, I just want to do a quick plug for joining us for our first roosting hours of 2025, which is coming up on January 30th. So roosting hours is an after-hours adult time to enjoy our facility. If you enjoy DraftWorks beverages, they've donated the beverages for that event as well. And the chance to hang out with your friends as well as our bugs and engage in seasonally themed crafts and trivia. I hope you'll join us for this increasingly popular event. You can check out our website for more information, and I think there is some information on the back table and as well as the check-in table. So tonight, to kick off the first Bugs and Brews lectures for 2025, we are thrilled to welcome Dr. Erick Greene. As many of you know, Erick was the former director of the University of Montana's bird ecology lab, and he was the guy that was always on the news whenever there was a story about osprey. Tonight, he will be talking about the game of hide and seek that goes on between predators and prey, looking at some of the astounding ways that caterpillars, moths, butterflies stay safe from avian predators. And I just have to say, his bio that I will read is probably one of my favorite bios that I've ever read, just for a variety of reasons. Erick Greene is Professor Emeritus in the Division of Biological Sciences and in the Wildlife Biology program at the University of Montana. He grew up in Quebec, Canada, with twin passions for music and nature. Erick dropped out of high school and lived for a year in the Galapagos Islands, working as a researcher on Darwin's finches. He then worked on seabirds 800 miles north of the Arctic Circle with the Canadian Wildlife Service. He returned for undergraduate studies at Dalhousie, Dalhousie University in Halifax, Nova Scotia, where he studied biology, music, and mathematics. He fell in love with ospreys in Nova Scotia and has been studying them ever since. He then received a PhD from Princeton University. He was the director of the UM's Bird Ecology Lab and has broad interest in behavior, ecology, evolution, and conservation. While studying insect-eating birds, he fell in love with the insects. He has studied the fascinating lives of some unusual caterpillars, spiders, and other arthropods. And with that, welcome Dr. Erick Greene.
Thank you, Glenn. Thanks, everybody. It's so delightful to see so many people on a really cold, Quebec-ish sort of evening. First of all, this is such a thrill. I knew Jen and probably 25 years ago when she was starting off at University of Montana. And even way back when, she had this, and she and Glenn had this vision of doing something like this. And everybody who knew her were like, yeah, right, well. And here we are. It's pretty amazing, so way to go. All right, this is meant to be a fun, informal talk, so please, if you have questions as we go along, just raise your hand or shout them out, because that'd be great. All right, so many people think of caterpillars as perambulating intestines, right? They don't do much except eat and poop, right? But what I'm going to tell you about tonight is, they are important food for birds, of course, which is sort of how the route I got interested in insects by studying eating birds that I'll tell you more about. But arthropods and caterpillars in particular are incredibly important food sources for many, many, many animals, including insectivorous birds. And tonight, what I'm going to tell you about, here's a little roadmap, because I'm going to sort of take you on a rather tortuous journey, but I'm going to talk about the really fascinating evolutionary arms race that exists between, in this case, insect-eating birds that are, and I'll be mainly talking about birds that are using their vision to find their prey. So this incredible arms race between predators and prey, and some of the most astounding things that have evolved out of this interaction. And I'm going to talk just a little bit about predator psychology and how it's important in the outcome of these predator-prey interactions in very interesting ways. And there's a really interesting observation. Why do things that are nasty tend to be very brightly colored? Okay, so we'll talk about that. And what are things that are good to eat? Some things that are good to eat also tend to be really brightly colored. You'd think that would be sort of a giveaway. They're very conspicuous. And I'm going to talk about two really fascinating forms of mimicry, Batesian mimicry and Mullerian mimicry, that have both arisen out of these interactions between predators and prey in cool ways. And then sort of to put a bow on it all, I'll talk a little bit about why, if you want to ID butterflies, especially in some places in the tropics, it's just crazy out of control. Okay, so that's sort of some of the ground I'm going to cover tonight. Okay, before we get into that, I'd just like to give a little shout out. Many people, especially in sort of suburbia, USA, have been, I'll say, brainwashed that this is what your lawn should look like. A monoculture of like true greeny sort of lawn. And this is how we should spend our weekends, right? Slaves to our lawn. But if you haven't seen it, I encourage you, if you're interested in this stuff, to check out this amazing book. I've got a copy of it here if anybody wants to look at it. It's called Nature's Best Hope by an entomologist, Doug Tallamy, who's a prof in Maryland. And he's got fascinating information about how just converting lawns like that to things that are more bug friendly can be incredibly important for the conservation not only of insects, but supporting a whole trophic system, wildlife in our backyards, and so forth. So really, this is really important stuff. All right, so before we get going, I want to just give a little sort of evolutionary framework about these interactions between predators and prey. And biologists have got this thing called the Red Queen hypothesis that sort of summarizes the thinking about how visually searching predators or other sorts of predators and their prey might interact. And it's taken from Alice Through the Looking Glass, and I'll read this because the words are small. The Red Queen insists that they run faster and faster. After a long period of exhausting running, Alice begins to complain. Well, in our country, said Alice, panting a little, you generally get somewhere else if you ran very fast for a long time, as we've been doing. And the Red Queen responds, a slow sort of country, said the Queen. Now here you see, it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that. And so this sort of encapsulates this idea of this escalating arms war between predators and prey, and adaptation and a counter-adaptation. So we might think of it this way, and I'll show you some neat examples of this. Let's say we have a predator that uses its eyeballs to search, and it's out there finding, let's say, caterpillars hid in the foliage. Well, this might put selective pressure for the caterpillars to be well hidden, to be cryptic or mimic something else and be harder to find. Okay, so there's sort of a counter-adaptation. And then that, in turn, might select the predators to evolve in certain kinds of ways, being better at looking for things, forming what are called search images that I'll talk about, learning, various forms of learning, and behavioral searching tactics, social foraging, and this can lead to sort of counter adaptations in the insects, and so on and so forth. So what I'm going to do is show you some amazing results that have developed undoubtedly from these interesting arms races between the predators and prey over a long time. All right, so I'm going to start off down in the Chiricahua Mountains in southeast Arizona, so on the Mexican border. This is where I did my PhD. And anybody been there? Oh yeah, look at that, yeah. Any non-birders been there? Yeah, okay. It's a really, really wonderful place. Insect people, you should go there too. After the monsoons, it's just crazy with insects there. It goes from low Sonoran Chihuahuan grassland yucca desert up through, this is oak juniper, all the way up through ponderosa and Doug fir, really interesting mountains. And I was studying foliage gleaning insectivorous birds. So these are generally little dicky birds that make their living with their eyeballs, finding caterpillars and arthropods hidden in the foliage. So here's a black-throated gray warbler. They might feed their babies, hundreds if not thousands of caterpillars they find. Okay, so that's their job. That's the only thing they eat. And as part of my research, one of the things I was interested in was trying to figure out what impact the foraging activities of these birds was having on the insects in the trees, right? If they, what they actually found and ate might be just a drop in the bucket. It might not really matter, right? And so what I did was construct a lot of these bird exclosures. They're a little hard to see, but it's a combination of Neolithic and space-age technology with satellite poles and agave stalks and stuff, but bird crop netting to keep birds off of one half of the tree. So each of the tree is netted, only half of it's netted off. So birds could forage in one half, but they couldn't get in the other half. And other sorts of predators like lizards and stuff could get in the netted off halves. So this was only keeping birds out. And what I found was that in many, if not most years, the birds were reducing the sort of standing crop of insects in the trees by 50% or more, okay? So these activities were having a major, major impact and undoubtedly exerting strong selective pressure on the insects that were living there. And so I spent about half my life as a student up in ladders in the trees sampling insects. And when I started, I really didn't know much about insects, but as Glenn mentioned, I just absolutely fell in love with the insects and ended up getting distracted and doing lots of side projects. And so here's, if you were a black-throated gray warbler, this might be your job to look through this foliage and find the caterpillars. Okay, there's 10 caterpillars hidden in this foliage, and I assume you've all found them. Okay, you all flunk as black-throated gray warblers. Okay, so I'm just gonna show you, this is sort of a rogues gallery of some of the caterpillars that I discovered when I was up in trees. And this will give you sort of a sense of the amazing adaptations that these arthropods, yeah, somebody's seen it back there. Okay, this plant is called alligator-barked juniper. And you can see the foliage is green with little white waxy chevrons and brown capsules. Well, hidden in there, and I've moved my camera, you can see there's a caterpillar hidden there, and it's a really, really good mimic of this foliage, right? It's got little white flecks on it, it's got a brown head capsule. It's a really good mimic. It's interesting, the systematics and taxonomy of this is in the family Geometridae. These are the inchworms. So these are the caterpillars that go like this. They walk like that. So the naming is done mainly on the adult forms of the moths or butterflies. And so I took some of these caterpillars to the leading world experts in this group, and they would say, don't know what it is, raise it up, feed it, get it to pupate, get it to hatch out, and bring me its genitals. and I'll tell you what it is. So my wife, Anne, back there, we did this work together, and she's an amazing caterpillar farmer. So we had a whole lab just full of peanut butter jars, you know, what you did as a kid with holes. And so we'd feed this and put in some fresh juniper foliage every couple days. And then one day I went to change the foliage, and this caterpillar was gone. It wasn't there. But that was impossible because the lid was still on, there was little tiny holes that couldn't get out, and it was gone, and I was totally flummoxed. The reason is, and I'll come back to search images later with birds, is that's my search image. That's what I was looking for. But as this caterpillar ate and grew, it got bigger and fatter, and it would no longer be as well hidden there. So when it molted, here's what crawled out. A brown juniper twig mimic. Okay, so I was looking for the wrong thing. It was there, I just overlooked it. And it's interesting with this quantum shift in morphology, going from a green foliage mimic to a brownish twig mimic, there was a quantum shift in behavior. It no longer went and hung out in the foliage. It only ever hung out, on the scaly brown twigs at the proper branching angle. So that was really neat. And then, if that's not amazing enough, it got bigger and fatter and molted. And it's a little hard to see, but if you follow down the twigs, these scales fall off and you get sort of a smooth pinkish orange stem. And so this thing unzipped its skin, and here's what crawled out. A bigger, smoothish, orangish juniper twig mimic. So if you and then it pupated and hatched out, and we figured out what it was. And so if you think about it, this is amazing developmental biology, right? There's genes controlling all this developmental biology, and this little perambulating intestine has, in its lifetime, has gone from mimicking foliage to a scaly twig to a smooth twig. Yes?
What was the time frame for those?
Oh, hi. The time frame was about 3 weeks. Yeah. Another neat thing, I digress, but the genus of this caterpillar is Cochisia, and it's named after Cochise, one of the last of the great Chiricahua Apache chieftains, and the U.S. cavalry tried to smoke him and his band of the Chiricahua Apaches out of these mountains, and they hid out in the junipers, and the U.S. cavalry couldn't find them. So I think it's a very cool caterpillar, and it's named appropriately after a great Chiricahua Apache chief and warrior. Okay, here's another neat story. This caterpillar is not particularly well hidden, but it's a cool story, and I'll bring it around. This is, it's got an ant on it, right? And mostly when ants find caterpillars, they thank you very much. They're butchers, they chop them up, nice protein, bring it back to the babies. But if you watch these caterpillars, there's ants always on them, and they ride shotgun on them. This is an acacia tree in the desert, and those caterpillars eat the acacia flowers. And this is in the group, the lycaenids, the blues and the coppers. We have, you'll see like around puddles or in the spring and summer here, little tiny blue butterflies. We've got a lot of in the same group here, and we do have ant-tended lycaenids here, right here on Mount Sentinel in the lupins and other plants. Okay, so I was photographing this neat caterpillar. There's two here you can see, and here's what's going on. You can see there's a caterpillar riding shotgun, but there's also a caterpillar tickling the rear end of, an ant tickling the rear end of the caterpillar, and they do it in a particular way, and you can see a little droplet of liquid. So, and the caterpillar is feeding the ant. When it's tickled in that way, it's got a special gland, and it's like a Coke dispenser machine. But it's actually not mostly sugar the way Coke is. It's got a lot of essential amino acids that the ants have trouble getting elsewhere. And so a lot of the nutrition of the ant colony comes from this interaction. All right, so the caterpillar, when I was photographing this, the caterpillar is eating an old acacia flower, right? But when I was looking through the viewfinder, I was having trouble focusing. Something wasn't right. And it turned out that the thing it's munching on is actually a caterpillar. Yeah, what? That's exactly. And it did such a good job of mimicking a flower, an acacia flower, that it even fooled an acacia flower eating caterpillar. And here it is, the caterpillar said, heck with this, and it ran up to the top of the thing there, and you can see its head and legs and stuff. But normally they would like curl up in a ball and look like a flower. All right, so 2 neat caterpillars. Oh, the one thing that what do the caterpillars get out of this interaction? They feed the ants, but the ants are always with these caterpillars, and if parasitic wasps or even birds come along, or lizards, the ants are all over and stinging them. So it's a protection racket for the caterpillars, and they get fed in return. Now, if that isn't cool enough, what happens when these lycaenid caterpillars get ready to pupate? They sing to the ants. They stridulate and they produce a sound that tell the ants they're ready to pupate. And the ants lead the caterpillars, a procession of caterpillars, down the plants, onto the ground, and they escort them down into their ant nest, way underground, and they have special chambers that they've dug called yeah, you can't make this shit up, right? And so here's a photograph I took of some ants with a whole bunch of these pupae of these lycaenid caterpillars, and this is underground in the dark in these, like 3 or 4 feet underground. And they turn them over, they tend them, they get dirt off them to make sure mold doesn't grow out of them. And then when these things are they hatch underground, and with their wet wings, the ants help them get to the top of their ant mound. They dry their wings on the outside of the mound, and then it's like a little volcano of blue butterflies flying off, and they go to the nearby acacia trees and mate and lay eggs, and the whole cycle repeats itself. So this is a very interesting interaction that's been driven by predator pressure, okay? And this is the way that this particular species has figured out to be safe. Okay, here's another really, really neat example of a caterpillar that my wife Ann and I spent quite a bit of time working on. This is an emery oak tree in flower in Arizona. They'll be like this in a few months. So the leaves are just coming out, and all those frilly things hanging off are the catkins. So those are the male staminate pollen-producing flowers, okay? And when I was watching birds, these insectivorous birds, I would see them occasionally grab a catkin and eat it. And I was like, what are these, vegetarian warblers? Or like, they're not supposed to be eating salad. And so I had no idea, and I was like 30 feet up in an oak tree looking at the catkins, and one of them walked away from me. And can you see the oak catkin mimicking caterpillar here? It's really hard to see, and in the next picture, it's right here. And to prove that it was there, I actually moved the caterpillar off the catkins so you could see it. And if I held one of these caterpillars in one hand and a catkin in the other, sometimes you couldn't tell them apart until one starts walking away, okay? That's how good they are. You can see they've got fake stamens up their body, and they're fuzzy and bumpy and golden brown. So they do a really, really good job mimicking the oak flowers, okay? So what did we do? We didn't know what it was. So we raised it up, and it hatched out, and we figured out it was a beautiful emerald green moth, and we have some of these in the same genus here. They're called emeralds in this genus Nemoria. And a couple months later, I was up in the oak trees, and I found a very, very different caterpillar. Okay, it's a mimic of this new first-year oak twigs, right? And later in the summer, all the catkins have fallen off the trees, right? And so we didn't know what that caterpillar was, so we raised it up, It hatched it out, and it turned out, much to our astonishment, to be the same darn species, Nemoria arizinaria. And so we spent a lot of time studying this, and here's what happens. There's 2 broods of babies a year. This is called a bivoltine, two broods a year. The early spring brood of caterpillars are these catkin mimics. But if you think, and then they mate and lay eggs, and their babies hatch out later in the summer. But if you think about it, if the caterpillars later in the summer turned into oak flower mimics, they'd be conspicuous as all get out because there's no flowers, and they'd get munched up just like that. So they turned into these oak twig mimics, okay? So pretty astounding, right? And so the developmental biology of this is really fascinating. There's genes that are getting turned on or off during early development to send these very tiny caterpillars along the appropriate trajectory so they turn into the right thing. Yes?
So do they switch every generation, or is it just when they're born?
So when they're born, they're almost microscopic. They're really, really tiny, and they don't really look like either form. And we've done a lot of work on this. I can talk to you about it later. It's about the second instar that genes are turned on and off to send them along this way. But what we got really interested in asking the question, what cues, what environmental cues are these caterpillars using to trigger the development to very different directions, right? And so what's the difference between spring and summer? What are some of the things that might, what's day length, temperature, humidity. Gosh, you guys are good. And what else? One other thing. So we have daylight, so photoperiod, temperature, humidity. Pollen, yeah, these guys, I didn't tell you, but they eat the flowers. And these guys, there's no flowers, they eat the leaves. So diet. So those were the 4 things that we thought, well, it's probably going to be one of these or a combination. So we did a lot of experiments to figure this out. And here's how we did it, just to show you how you do this research. You get some slave labor, unpaid slave labor in the form of your daughter. So this is our daughter, Robin. Out at night, you put up these black lights, which now Jen and Glenn have. And you can see, so black lights are, they emit a lot of UV, like those bug zappers. Do not use a bug zapper. 99% of what they kill are beneficial insects. But you can see this isn't even craziness. When the monsoons come, you couldn't even see the sheet. It's so covered with insects. And I've had sheets, that's a bed sheet that's just tied up. I've had so many insects on the sheet that the sheets have ripped off and fallen on the ground under the weight of insects. But Robin is looking for females of Nemoria, and when she finds one, she'll put it in a little vial. Here's why they're called emeralds. So this is the adult moth, beautiful emeralds. And so they lay a whole bunch of orange eggs, and then when the little caterpillars hatch out, we can divide up the siblings into different groups and rear them under different conditions, okay? And so we reared them independently, varying everything you mentioned: photoperiod, temperature, humidity, and diet. Okay, what do you think is important here? Okay, you rear them on catkins, you feed these babies catkins or oak leaves. 100% of the ones that eat pollen turn into the catkin mimics. 100% of the ones that eat oak leaves turn into the leaves. The other things don't matter. They don't influence this at all. So this really is a case of you are what you eat, okay? It's entirely what we call a diet-induced developmental switch. Okay, so that's pretty cool. So this is very sophisticated developmental biology with complicated genetic architecture that is a result of this interaction between birds looking for them, right, and how their genes and developmental biology work. So pretty crazy. this was this was published as the cover of Science magazine, which is a pretty good science journal, and one of the best science journals in the world, I've been told. And I did this when I was a student, and then went and got a postdoc at UC Davis. And there had been, when this was published, there was a lot of places that were interested, so it got picked up by, you know, National Geographic and lots of places. And then when I was at UC Davis, I got a call from a writer at National Enquirer magazine. And he said, like, I'm doing this story on these here caterpillars. And this was at a time when the National Enquirer was publishing these things about science under the banner headline, "Look how your tax dollars are being wasted." And it was a very scary time for science. And scientists who were being funded by, say, National Science Foundation or other places were being called in front of Congress because some person had read something in National Enquirer magazine, and they were having to get lawyers and protect themselves. So I was like, I'll pass. I don't need you to write up my thing. And he said, no, we're doing it not under look how your taxpayers' dollars are being wasted, but under what we call creature features. Isn't Mother Nature wonderful? So I said, okay, I can live with that. So I told them this story. You know, you feed them this, they do that, blah, blah, blah. And then a few days later, I got a call back from a different person who was his boss. He said, do you expect us to believe this? and he said, this is like, you know, this is just too amazing to believe. Like, you have to tell me some people I can talk to verify this story. And I was flummoxed, and so I gave him the chair of the biology department at UC Davis, the chair of the entomology department at UC Davis, and the chair of evolution at UC Davis. And then a few days later, I got a call back from like the whole head chief guy. And he's like, okay, we're on to you. The names you gave us, they're all people at your institution, so it's clear they're in on this scam too. And so I said, I don't know what to tell you. I mean, who checks your stories on Elvis weds nun, fleas, and UFO? And at that point he said, oh, we're a reputable news journal. You're thinking of The Sun. And I said, I don't know what to tell you. It was published in Science, and you know. And he said, oh, it was? Oh, okay. And so they ran a story in National Enquirer magazine about this caterpillar, and on the strength of that, I got a job at University of Montana. It carries a lot of weight. Okay. Yes?
If you fed it a different food other than those two, which morphology did it?
Oh, that's a great question. I've done that. These are specialists. If you look like an oak flower, you better be in an oak tree. And if you look like an oak twig. So these are extreme specialists. You'll only ever find them in oaks. And we did those experiments. We fed them plants, common plants from around the area. They don't even recognize anything else as food. They would starve to death rather than eat something else. Okay? But the really interesting question, we have one around here that's pretty common, an emerald. Look for a little emerald in the summer near your lights. It's what we call a generalist. The babies eat like 20 different kinds of plants. And what's really neat is if you feed them those 20 different kinds of plants, they turn into different things. It's not like a perfect catkin mimic and a perfect twig mimic. It's sort of more of a gradient. So diet still is important. Yeah, so great question. Okay, I'm going to talk to you about another really interesting thing having to do with moths. And these are underwing moths. The genus is Catacula. We have them around here. They're called underwing moths because they normally hide during the day on tree bark. and with their forewings folded up, so they look just like bark. But if they're disturbed, they flash these bright underwings generally, and that's a startle response, and sometimes predators sort of are scared off and these things fly away. What's really interesting though, massively polymorphic, all that means is if you look at one species and you get 100 individuals and you look at their forewings, there's huge variation among different individuals in the same species. They look very, very, very different. They all look like bark mimics, but they're different. And so people got really interested in that because in a lot of species, you know, like a robin looks like a robin, looks like a robin, right? But there's a lot of variation in these things. And so people got really interested in why that might be. And so I'm going to talk to you a little bit about search images. So when I was looking for the green twig morph of Cochicea, I wasn't looking for a brown twig morph. I completely overlooked it. Here's some really cool experiments that have been done on this. Long time ago, before there were things called computers, the researchers did things like this. These are really great experiments. So here's a trained captive blue jay, and it's in a cage, and there's a little plexiglass screen, and it's being shown a slideshow. Okay, there's a tree trunk there, and on that tree trunk, there is either a catacala moth hiding or not. Okay, and so this blue jay is hungry, and it's been trained. It's looking very carefully. You can see at the tree trunk, if it sees, if it thinks it's found a catacala moth, it's been trained, it pushes this button, and if there was a catacala moth in the picture, a mealworm comes down the chute and it can eat, okay? What if it makes a mistake? If it pushes the button when there's no moth there? Well, this bar is wired up to like 5,000 volts of, no. The blue jay is penalized, so if it pushes the button and says, I see a moth and there's no moth there, it has to wait like a minute before the slide goes on to the next one. So it is hungry and it's waiting around. So very quickly they learn, if I don't see anything, I just don't do anything, and boom, it goes on to the next slide, and so that's how they get the most food. And here's the results. So here's one of the pictures in one of the papers. Here's an actual photograph of a tree trunk. There's 10 catacala moths hidden in that, okay? Like there's one, right? And there's 10 in there. So you get the idea. These things are really well hidden. But here's the interesting part. So I'm going to show you just a few graphs. Let me walk you through them. So this, with one blue jay, it was shown a first moth, okay? And so here's on the y-axis is the percentage of correct finding something that it was there. And so the higher up on the y-axis, the better they are at finding the well-hidden moths, okay? So if they're shown just a sequence of one type of moth, so it's the forewings are all the same, you can see really quickly by the fifth slide, they're basically batting 100, right? They're finding every single hidden moth, okay? And you can do that with another kind of moth, but if you just show them the same sequence, they get really good really fast. But here's the interesting thing. If you mix them up, they're looking for the wrong thing, right? They're looking for the last thing they've seen. And it's basically a coin flip whether they find them or not. So this was some of the very first experimental evidence that showed how these search images are really important in predators. And we know now from a lot more studies that most animals form search images. So they've got something that they're scanning the universe for to try to find what they think they're looking for. Search images are great. If you've got the right search image, you're more efficient at finding stuff. If you've got the wrong search image, you just overlook stuff, right? And this is the modern version of these experiments. So the same lab is now giving captive blue jays, they're letting them play video games. And here's a very famous blue jay, but same sort of protocol. You can see there's a button, and now the food comes out down below. But if it sees a virtual moth on the virtual bark, it pushes the button. Okay, here's some of these virtual computer game moths. You can see they're kind of funky. Here, and that's on a gray background just so you can see them. Here they're on background, but so they were sort of trained up, and by the end, those exact moths are hidden in this bark down here. Those things are down there. So this is the task the blue jays were asked to do. Find these highly cryptic moths on this virtual background, okay? And so the video games were they got to look for lots and lots of moths, and then the ones that they found and ate, they threw out of the game. The ones that were overlooked by the blue jays, they allowed those moths to have virtual sex and create virtual babies, and that would repopulate the next generation of the game, okay? And they had rules that made sense about if two moths mated, what their babies look like and stuff. So this is the last complicated graph I'm going to show you, and it also demonstrates search image. Okay, I wish they'd done this graph differently, but on this axis, they've got this weird thing called dissimilarity. Okay, I wish they said similarity, but dissimilarity. So if something has a low dissimilarity, it's like a double negative. It means they're very similar, okay? So, yeah, why don't you just say similar? So, for example, if the sequence that the blue jays found, they looked for this, and then the next one was identical, these would be a low dissimilarity, right? Okay? And let's say they were shown two things that were very dissimilar. They would be up there, okay? And on the vertical axis is the accuracy, how good they were at finding them, okay? And you can see a really strong relationship that when they were very similar to each other, they were really, really good at finding them, okay? And as these things grew more dissimilar, you know, they were getting, again, down near a coin flip. It was 50-50 whether they found them or not. Okay, so this also demonstrates the search image by these predators, which is super important, part of the psychology of like lots of animals. And what these experiments showed in this computer game is that overall, in the population, whatever moth population they started with, by the end of the game, the crypticity, how well hidden the moths were, got better and better and better and better, okay? The blue jays really made this population of moths get more cryptic. But what was really interesting is if you think about it, if any of these well-hidden moths got too common, like let's say this form was super common, then the blue jays are going to be making search images of them, and they're going to be really good at finding the more common ones, right? And they're going to overlook all the rare ones. And so what ended up with these experiments, and what undoubtedly happens in nature in these sorts of situations, is that what's called phenotypic diversity, and just think of the diversity of moth coloration patterns, it increases over time. And it makes, and this is called rare morph advantage. So the really uncommon things get overlooked by predators and they persist, and if anything ever gets too common, gets mowed down by the predators. So this is undoubtedly a really interesting form of selection by these visually searching predators that in some cases produces what is called this massive polymorphism. Okay, so sort of a different outcome from this evolutionary arms race. Glenn, how are we doing on time? Good? Okay. This is to remind me that there are more ways to skin a cat or to avoid being eaten by birds. And you can be like this. Like, you don't have to be well hidden. You can be nasty as you want. This is in the silk moth family. This is called the buck moth. Their host plant is oaks. And you just looking at that, you're not going to want to grab it. And here's a close-up I took of those spines. They look like hypodermic needles, right? That is because they are hypodermic needles. If you were to touch that, or if a bird was to grab it, those little needles break little bags of chemicals inside the hypodermic needle, and those are forcibly ejected either into your skin or into the mouth of the bird. Okay, and those, if a bird were to try to eat this, it could kill them. Okay, so yeah, so pretty neat. Here's just another couple cool ones. In the tropics, where there's a lot of really nasty snakes, this is an amazing snake mimic caterpillar. And that's a caterpillar. It's amazing. And this is, there's very, very dangerous pit vipers. That look just like this. This is a Bothrops mimic. So like if one of those snakes bit you, you would be in big, big trouble. This is a caterpillar that looks like a snake. I mean, there are bird poop mimics, okay? This is a caterpillar, and there's not too many things that like to eat bird poop. And I just found this photo. Check it out. This is mind-blowing. Here's a moth that has like a bird poop on its body, and it's got two flies on the sides eating the bird poop. Whoa, I know. So all these interesting, weird things that are out there in the insect world are interesting results of this Red Queen idea of this, this arms race. Okay, right now I'm going to switch gears again and talk about really bright things and sort of start with the question, why are poisonous things, why do they tend to be brightly colored? So I'm sure most of you know about the story about monarch butterflies and milkweed plants. So here's a monarch butterfly, and those are some milkweed beetles. And they're all really brightly colored, right? The beetles are bright red and black. And milkweeds are full, if you break them, they've got that white, gummy, nasty stuff. It's full of cardiac glycosides. Okay, so the milkweed plants themselves are well protected with these very, very dangerous compounds. These insects have figured out biochemically how to sequester them and deal with them. So they obviously don't kill them. But, and then what happens is when the monarch caterpillar pupates, it keeps those poisons, the cardiac glycosides, inside the pupae, and the adult hatches out with those poisons in its body. And so the adult monarchs are highly toxic. And some of them, if a bird ate some of the really more toxic monarchs, they could have a heart attack and die. I mean, it's bad news. They're well protected. Okay, so, but if you're poisonous, why be bright? Okay, like why advertise that you're there? And so the first really good suggestion about this was from this very interesting, important biologist, Alfred Russel Wallace, who lived at the exact same time as Darwin, and he actually wrote Darwin's book before Darwin wrote it, and he sent it to Darwin, and Darwin got scared and published his book really fast. And he wrote about this, that these things, like monarch butterflies, need some outward sign of distastefulness. It's necessary to indicate to its would-be destroyer that the prey is a disgusting morsel. And he talked a lot more about the psychology of predators, that having something really, really bright and conspicuous is going to be more easily learned by predators, okay? And it reduces what we mean by recognition errors that are talked about. So here's a poison arrow frog and another interesting silk moth caterpillar, and these are highly toxic. And if you think about it, let's say you had really dull, boring stop signs. Like psychologically, it'd be hard to remember this, right? Might end up with, you know, like, who's going to pay attention to a boring stop sign? But if you have really, really bright warning colors, psychologically, we know from lots of experiments that these sorts of things are easier to remember and recognize. And so that's the thinking on why highly toxic things, including lots and lots of insects, are what we call, have aposematic coloration. Really, really bright, contrasty colors. So here's two poison arrow frogs that are really poisonous, a coral snake and some poisonous beetles. And aposematic is from, we'll talk about some etymology. Did I get that right, Glenn? So aposematic is from Greek, and it means like back away and nobody will get hurt, basically. It means go back. And we now know that the evolution of these bright aposematic, easily remembered color patterns are related in another interesting way to predator psychology, and it has to do with what we call learning curves. And so imagine you're trying to learn some sort of task, and this is the first time you've listened to your language tape or something, okay? I know they don't have tapes anymore, but bear with me. And the second time and so forth. And on the vertical axis is basically how good you get at that task, okay? So if something is very hard, it's going to take you a long time to learn it. Like if you're trying to learn calculus, you got to be exposed to it a lot, right? Something that's a little easier to learn, you're going to learn it more quickly. There's a really interesting kind of learning, and we have it, all vertebrates and a lot of invertebrates have it. It's called one-time learning. One-time aversive conditioning. And that is, I mean, wouldn't it be great if calculus was like this? Like integrals, Spanish, like I heard it, I'm good. So this is like one exposure to whatever it is, and you got it, you figured it out. And we have one-time aversive conditioning learning, and it's typically associated with food poisoning. So how many of you gotten really sick and spent like a bunch of time barfing? Okay, what did you eat?
An egg that I didn't know was old.
Okay, and how did you feel about eggs after that?
It took me a while.
Yeah, okay, a bunch. What'd you eat? Yeah.
French fries. I got the flu after French fries. I couldn't eat them for 5 years.
Oh, yep. Okay, that's what's going on. We know that neurons in your brain fuse and like, French fries? Like, I'm good. Yeah, I had this with oatmeal. I used to love oatmeal when I was a kid, and my mom served me a big batch of oatmeal, and it wasn't the oatmeal, I got flu or something, and I spent like a long time being really sick. And my wife loves oatmeal, and it took me about what, like 30, 35 years before I could start to eat oatmeal. Yeah. What's that? Yeah, no. But it's interesting too. It was mainly the texture. If it was like a multi-grain thing, then it had texture, I was fine. But if it was slimy, like, yeah, in my house we called it vile glop. Yeah. So we know that this happens in predators, okay? And so if you think about these highly toxic things, and there's going to be, every generation there's going to be naive predators coming along, right? Like little baby blue jays. It's a complicated world. How do they know what's good to eat and what's not? They have to, sample, and there's going to be some learning errors, right? they're gonna like, hey, what's that? And they're gonna pick it up and chew it and like, don't like that. But there's gonna be some of the things that they sample that will probably die. Okay, here's a really neat case of this. Here's another captive blue jay, and it was raised from an egg. So, and it was completely naive about food, and the people who had it, they would just, with a sweep net, get bugs and stuff. And they were doing an experiment, so they were just throwing lots of stuff in there that was good to eat. Okay, and it was like, oh, great, here's the buffet, and it would just eat everything they gave them. And then they threw in, this was pretty devious, they threw in a monarch butterfly. It went over, thank you very much, grabbed it and ate it, and immediately threw it up. And this was work done by Lincoln Brower, a very famous biologist, who he was helped discover the overwinter roostering sites of monarch butterflies in the mountains in Mexico. And he devoted his life to studying monarchs. And this is a very famous photo called Brower's barfing blue jay that is in just about every animal behavior textbook. And so this was one time aversive learning. This bird never touched anything that was orange and black and white again the whole rest of its life. Okay? So it was good. Like oatmeal, monarchs, I'm good. I'll pass. Okay, so that's sort of the psychology from the predator's side that drives some of this stuff. But when you get this very bright, conspicuous stuff that offers such strong protection against predators, it opens the door for counterfeiting. Okay? So, you know, one of these, Rolex and the girl with the pearl, one is the real one and one's a fake, okay? And it's really hard to tell. And so when we look out there, especially in the insect world, we see some really, really amazing examples of this counterfeiting, of mimicry. And I'm going to tell you about two different kinds. Okay, so here's the first kind is called Batesian mimicry. And when a yummy species that's good to eat mimics something that is not good to eat. Okay, and this was named after William Bates, who was a British biologist, and here he's getting roundly scolded, rightly so, for he's shot a toucan and all the toucan buddies are giving him a hard time. But he described and figured out a lot of this early on. And this example is, and we've got this around here, monarch butterfly, which you now know is highly toxic from these cardiac glycosides. The viceroy butterfly is a bird poop mimic, and it feeds on things in the willow family, so like cottonwoods and willows and things. And there's no poisons in those, and they're perfectly yummy, but you can see that the adults are really good mimics of the monarchs. And so they slip under the protective umbrella of the really poisonous thing, okay? So this is called Batesian mimicry. And now I'm getting to the part of why is butterfly ID so difficult in some parts of the world. So here's one species of swallow-tailed butterfly. It's the African swallowtail. This is all the same species. These are all African swallowtails. And here you can see there are non-mimetic forms, so they've got nice respectable swallowtails. All of these other ones are mimicking, they're all papiliodardanous, but all of these different forms are mimicking other species of toxic butterflies. And there's actually more than 14 that I haven't shown you. So in one species, different individuals can be mimicking different models, toxic models, okay? Crazy. These are all African swallowtails. I mean, you'd think if you looked at it, it's like they're all different. This is one species, and they're mimicking different things. Okay, I'm just about to wrap it up. I see some yawns here. This is another really, really interesting kind of mimicry. So here's two different species. So these columns are two different species of what are called heliconius butterflies, and they feed on a vine that is highly toxic. So these are like our monarchs. 2 different species, both of these are poisonous. Okay, but if you travel around South and Central America, you can see that in different places, they look very different, right? So this is Orado. You can see that if you're in this part of the Amazon basin, they look like this. If you're up in Ecuador, they look very different, but these are the same species. So what's going on here is this warning coloration, the stop sign, is kind of arbitrary, right? It doesn't really matter what it is, as long as it's really bright and conspicuous. So there's geographic, there's geographic variation in the warning color pattern as you travel around. But what's really interesting is that 2 different poisonous species have converged. When they're living together, they look the same, right? So they're sharing the same stop sign. And what we know now is that it's because of these the sampling recognition errors. I'm almost done, Glenn. No. Yep. And if you think of, no, you're good. And so if you think about it, if a naive predator had to go and sample, say, 20 or 30 poisonous things that looked quite different, there'd be a lot of mortality from those naive predators just learning. But if all the toxic things share that burden, they all look the same, that like all of a naive predator has to do is like try one, and it's like everything that looks like that is protected. So it's thought that there's this convergence in protective coloration to sort of reduce that burden of mortality from naive predators. Okay, so this is called Mullerian mimicry, and it's named after Fritz Müller, who was a really interesting German guy. He applied to medical school in Germany and was rejected, and he was so depressed, he got in his pajamas and wandered around in the jungle in South America. And thank goodness it was biology's gain, medicine's loss. He did a lot of work on this really interesting stuff. Let me just show you how complicated it gets. When you go to some places, so these are everything above the lines, these are all poisonous. These are different species. So you can see here's 1, 2, 3, 4 species, they're poisonous. So these are Mullerian mimics. And then these things that live in the same place are good to eat. So you've got, in the same place, you can have some good to eat Batesian mimics that are slipping under that protective umbrella, and poisonous Mullerian mimics. And then I'll just end up, if you go in places in the Amazon basin, and I've been here trying to figure out some of this stuff, there's what they call mimicry rings. So here's what's called the orange tip ring. Here's the dark transparent ring. Here's the light transparent ring. Here's the yellow bar tiger ring. These are only 4. There's at least 4 other common mimicry rings. The amazing thing about this is that there's typically more than about 120 species in each of these rings. So there's 120, if you go down and see something like that, like good luck figuring, there might be 120 species in that area that look almost identical. And some are toxic, so those would be the Mullerian mimics, and some are good to eat, but they look like that. Those would be the Batesian mimics. So this is just to give you an example of how sort of crazy these predator-prey arms races have been and continue to be in the living world. And so I've just sort of scratched the surface to give you some neat examples, and I hope you walk away with an appreciation of some of the really cool things that caterpillars and moths and butterflies have evolved to deal with predation by visually searching predators. Thank you.
time for questions if anybody's got questions. I'm sure he's got answers.
Has mimics ever been so successful that they tilt the balance to gain in efficacy and then the poisonous ones
So the question is, have mimics ever been so successful?
so successful that they've tilted?
Yeah, that's a great question. The question was, have mimics ever tilted the balance to make things unstable? And I guess a way to think about that is, imagine there's a few Mullerian mimics, so they're bright and they're poisonous, and then you get more and more species, right, copying that. So there's mainly things that are yummy to eat. So at some point, that protection is going to be weakened because young predators are going to be more likely to run into yummy things. And so one of the things that people are really interested in that question. And that's probably why there's so many mimicry rings, is that when they become unstable, things go to something else. We don't know, but it's yeah.
Can you touch on the very basics of bird vision versus human vision? Like, what is I'm curious about what a bird.
Yeah, it's a good question, which is, can I talk about bird vision versus our vision? So we have 3 different color receptors. Birds have 4. And the 4th one is a UV color receptor. So they can see more like honeybees. They can see stuff down in the ultraviolet. So all these pictures were taken with just, things that we can see. We know that probably some of these things are just crazy if you included the ultraviolet. So some of these caterpillars are probably, and moths are probably even zanier with the UV component, but birds can see that. So their vision is kind of better, certainly in terms of color vision, because they cover a wider range of the electromagnetic spectrum. And you've probably seen pictures of flowers taken with ultraviolet. So some flowers don't look all that snazzy to us, but if you take a picture with ultraviolet, it's like they're signaling to the pollinators, restaurant open, restaurant open. Yeah.
Is the only way that the predator can decide if something is poisonous, is it has to do with trial and error, or can any of that be passed on?
Yeah, so good question. The question was, can predators, can they only figure out what's good to eat by trial and error? And can that be passed on? For a lot of probably birds, they have to learn it on their own. But more social birds that stick around as families, we know that they learn a lot of stuff from their parents. And so things like crows and really intelligent things that form family units, we know that they're teaching their kids what's a predator, and so for a lot of birds, they probably have to learn it themselves, but some highly social birds that stick together in groups. And you just reminded me of something that was really interesting the other day. We've got, there's a gazillion dogs in Missoula, right? And the crows around our house never, ever pay any attention to the dogs, right? They're just dogs walking down there. And the other day I went out and the crows were going nuts. And I thought they had a great horned owl because they don't like owls. And they were just like 20, 30 of them just going crazy. And I went out and looked and there were 2 foxes right in the middle of the road, right in town. And the crows distinguished them. They knew they weren't dogs and they were bad mojo. It's like, that is a predator. And so somehow they know that, and there were young crows in the flock, and they were getting that information socially, learning that whatever that is, it's not a dog, and it's bad news. So yeah.
How much hybridization is occurring between these mimic or these breed groups? Like, our perspective, it looks like they're the same species.
Yeah.
But are they distinguishing amongst themselves, or is there crazy hybridization?
Yeah, so the question is, do some of these mimicry groups hybridize? And I don't know the answer to that, but like some of those, the heliconius butterflies, they're in the same genus, and they could be hybridizing. A lot of insect mating is through chemical pheromones and not visual stuff, and the pheromones tend to be quite different between even closely related species. So I would suspect there's not a lot of hybridization, but I don't know, maybe.
There is with some California species.
Okay, yeah.
But yeah, the pheromones kind of sorted.
Yeah, so it's sort of out of my area, but yeah. Pat?
This arms race is a result of evolution over how long? What's the kind of time frame for one of these things to develop or change?
4,624 years. Yeah. I'll talk to you later. One more.
So why do some of the caterpillars not develop that mimicry and instead go for like, one of the examples you showed, a bird poop mimic, and that developed bright coloration later as an adult? Why not have that bright coloration in both phases?
Yeah, good question. I mean, it just shows that there's many end results of these interesting interactions. And so in the case of the viceroy, you know, the caterpillar, I mean, it could mimic a toxic caterpillar. You know, why doesn't it look like a monarch butterfly caterpillar? It doesn't, it looks like bird poop. Great question. It probably, it's a different way of doing things, and it's really effective. There's not many bird poop specialists out there. And yeah, but it's an interesting segregation of different life stages, right? All right, thanks, folks.
Thank you so much, Eric. That was awesome. Just as a heads up, the next Bugs and Brews will be February 25th. Matt Seidensticker with the Northern Rockies Research and Educational Services will be talking about the Montana Moth Project. And tickets for that will likely go on sale